thing, and I was telling Panda yesterday that when we were landing, it was amazingly green. I mean, coming from California, that's not this kind of an unusual sight. But by the way, California is quite green now, in case if you haven't been there in the last few months, because of all of the rains that we had over the past 2-3 months. The hills are all, I've not seen that like that for pretty much 30+ years that I've been there. So, yeah. So it's just, I think rains do good things sometimes. So I'm going to be, so this is a kind of an unusual talk for me because my main intent was to talk to many of you on the Bisque platform. So I kind of put together things, bits and pieces. So you might find it a bit disconnected. It's not my traditional research talking about a particular computer vision problem, but focus from the cyber infrastructure perspective. So if you have any specific questions on any of the topics, we could take that offline, or if it's something that's easy for me to answer in real time, I'll be happy to do that. Okay. Why is it not moving to the next slide?
You need to click on the Zoom part, on the slide part of things. Right now it's on Zoom.
Because it's not in the. Yeah. I bought this for this meeting.
No. Just click the mouse on the.
Yeah, just click.
On the anywhere.
No, yeah, perfect. But this one doesn't work.
It will now.
Oh, it will now. Oh, thank you. That's news to me. I'm learning too many things today. It's talking here.
That's you.
That's you. Oh, that's me. All right. So I should acknowledge my sponsors, some of our ex-sponsors here in this room, the NSF, many of my students and collaborators, and I'll try to acknowledge them as we go through this. And this is the only slide that I'll have for my own research group. So we have quite a few graduate students. In fact, I prepared this about a year back, and then I realized that all of these students are mostly gone. They've all graduated, many of them. But one of the things that Bisque has enabled, and I should point this out here, is bringing in undergraduates, high school students, and graduate students today. So in fact, during the pandemic time, this was the only way to kind of collaborate with the data. So that was, I think, was a great experience. In fact, I think one of the papers that are coming up that will be published soon will have a couple of high school students as well. So we have the first clinical paper that should be appearing in Neurosurgery Journal sometime in June on one of the topics. The purpose of this slide is to kind of essentially so we have a whole range of activities here. The pointer for some reason is not working. I think it goes to the notes, but not the one. That's okay. I can. So we have students who are working on the biomedical data, microscopy imaging, all the way to subject data coming from MRI, fMRI, CT scans, and MRI scans. We have a couple of students working on the environmental related stuff. In fact, the CVPR paper that will be presented next week on the what we call, and I'll be talking about it in a little bit more detail, on wildlife monitoring. And then we have, I'm not going to talk about the security forensics part, but that's a very interesting direction that we have taken, where bringing in computer vision methods to work on detecting computer viruses. So if you're interested in that, then I can easily talk on that for a couple of months, but it will be a distraction. And then we have quite an amount of work over the last few years in terms of materials related applications, and I'll probably point to a few of those things. The bottom line here is that these are multimodal data, multidimensional, unstructured, sometimes time evolving, etc. So to create a usable platform that would help us share, collaborate, analyze. And you probably will hear me repeating these things again and again throughout this presentation. And this morning I inserted this slide from our site visit on our Center for Bioimage Informatics and NSF funded ITR project back in 2006. And this was a slide that this is being hidden here by this. Let me move that out of the way. So Steve Fisher was one of our early collaborators on the bioimaging side, microscopy imaging side of things, and he put this slide together for the NSF site visit. Yeah.
The Zoom people are saying they cannot hear you very well. So maybe we can use that mic. There's a mic there. Oh,
No, no, I can hear you.
okay.
Okay, I'll stand. Not move. Hope you can hear me now. Okay. All right, so you see that I'm not going to read out all of the stuff here, but pretty much whatever that was said back then, you just scale it up by maybe a factor of 10 or 100. All of those observations are still true today. Each of these projects, individual PI projects, generates instead of thousands, maybe hundreds of thousands of these images. And by the way, they were storing these things on micrographs, literally printed once. Now they're scanning them and they were kind of trying to scan them and see if they can digitize them.
Recording in progress.
And there's no convenient way to efficiently organize, share, or search. And for most part, this is still true, right? I mean, we have made some inroads into this, but this is still a major challenge when it comes to multimodal data. And how do you let others access it, work collaboratively? All those things are still significant issues.
Just click again on the slide, anywhere on the screen. Just click anywhere.
So this is really multimodal interaction, multi-device interaction. So there is a need to develop an infrastructure. There was a big need to develop this infrastructure, and that's what we try to do. And in terms of scalability and reproducibility of the results, being able to robustly run your methods and organize your data, share the data, as I already said, but you can also share the methods. And provide some level of interactions. So unlike the experiments that you see today with ImageNet and stuff like that, where you hope the primary goal is to collect millions of these images and train a model, many of these images are individually like gold pieces. You really, I mean, very high quality, high valued data items that you want to preserve. It's not like I'm collecting 10,000 images in one stream. So how do you interact and provide whatever resources that are necessary to analyze and manage them? So that was, so even though the project was, the original intent was not to develop the infrastructure, we ended up putting quite an amount of effort into what is now referred to as Bisque, which is open source. You could run this as a service, and you could download it and set up your own infrastructure for that, or we have a version of it that's available through UCSB and till recently through Cyber's infrastructure, where you could log in and use whatever the resources that they provide. So most of my topic will be focused on the Bisque part, but this is one slide I really want to bring up here. Part of my presentation a couple of years back at a workshop in Europe, and then I actually borrowed this from where I have the reference back there, right? So much of the work prior to the 2000s, before the deep learning and all of that stuff happened, right? We used to have the traditional signal processing, information processing part of it, and you would work with domain researchers to do something interesting, whatever the computationally interesting part. So that's kind of what I would call traditional research, right? But then the data revolution, and then the machine learning thing happened. And then the collaborations kind of became basically within electrical engineering, signal processing, and computer science spaces, but maybe with a little bit of data that you borrow from other sources, right? Like ImageNet is a good example. And I like the part of the danger zone there, where you think of this as not information processing, but somehow magically you can discover things. I think that's the trend we see much of the time today, without really getting deeper into the science domain problems. And I think that's the part that we should try avoiding. And maybe, I mean, this is the sweet spot in terms of the intersection that I would like to call data science. But I think for most part, any of these intersections these days is broadly speaking data science. But there is a danger in, particularly if you're in science applications, not giving much attention to the data aspects and the science aspects of it is going to be an issue. Okay. So coming back to this, right, organizing, sharing, etc. These are all challenging problems. You need reproducible and robust analysis workflows that can be engineered. And the real problem, which relates to the previous slide, is that each problem is going to be slightly different. How do you enable transformative changes with that? So this is the kind of a very first one slide introduction to what works with. Today, if you use this, these are the things that and we will go through these in much more detail in the remaining slides. It provides you a basic platform to share, store your data. And the way I try to explain this for those who are not familiar with this, think of Google Docs. So this is Google Docs for multimodal. However, in addition to Google Docs, what Google Docs can do, you can also bring in your analysis. So that's a very probably a simplistic way of thinking about it. And more importantly, it is not data specific, it's data agnostic. So as you will see, even though we started this project as a platform to support microscopy imaging data, we now have applications all over the spectrum. I probably have the slides coming up where I talk about specific applications to me. So it's not specific to the imaging problems that you're looking at. It could deal with 2D, 3D, 4D, 5D images. And out of the box can support over 200 different imaging formats, pretty much most of the existing microscopy imaging formats. It could support many of the GeoTIFF and variations and medical imaging formats such as TIFF. It can support tables and similar stuff. The other stuff is being worked on. Depending on who is interested in it, we could make those things available. But right now, there hasn't been much of a significant effort on that. And then one of the things right from the beginning we wanted to emphasize is that everything should be web accessible. So the only thing on the client side you should have access to is a good web browser. So there's nothing to install as long as there's a service that's running behind the scenes to support. And it just easily should be scalable. So right now you can take this and deploy this on Amazon in an hour or two. As long as you have a good credit card to support the expenses, you can get this thing up and running fairly. And again, essentially repeating what I said in the previous slide here. So it provides the core infrastructure, provides the ability to do the management, the storage part of that. Right, and it's just drag and drop kind of a stuff. You can easily organize this according to your preferences, and then you can do the visualization all within the web browser. I think we were probably the first ones to engineer a 3D web viewer. So if you have 3D data, you can actually kind of nicely visualize that. In fact, some of those elements were adapted by various groups, including Allen Cell Science Institute. So if you go to the what is it called, cell profiler or something like that. In the back end, Bisque is the one that is supporting the 3D parts of it. So use cases. So what I'm going to do next, and please feel free to stop me at any point in time. Okay, I'm going to go through some of these examples that I have here. Kind of tell you a little bit more about the applications and also relate to the corresponding research that we are doing in the lab. The first one is, so this group, MARE, it is a non-profit organization that's kind of has some connections to UC Santa Barbara. What they do is kind of illustrated on the right-hand side here. They actually have these UAVs that they can navigate very close to the surface of the ocean and take videos. And they are interested in classifying many of the invertebrates that one can find. And the way they do that currently is, you see that you have the video that's kind of, as the platform is moving forward, there are two laser guides there that kind of tell you, you can, from those things, you can get some additional measurements. We are not using that explicitly. You can, what they do is they actually, so if they're interested in counting a particular object of interest, so let's go to the next one here. They literally, the human experts click, they do the mouse click to count the objects. And you can imagine how laborious that process is. They have thousands of hours of video that they have collected off of the coast of California, Southern California specifically, and in Florida and in the Caribbean areas. But that's their problem. And we don't have a good solution, but we have a solution at this point that would at least help them do this in a better way. So this particular part that you're seeing is already available in this. So for this project, the particular innovation we had in the most recent version of Bisque update is to be able to ingest a video as opposed to just images and image sequences. So you can have an image view where you can do the annotations on the videos. And you can play those videos, and you can, as you saw, this is the output that was generated by the Bisque module that is running behind the scenes. So you can do the detection, tracking, counting of these things, and actually producing a summary results at the end. So that's something that one can do today. Some of these objects, I don't know whether you had time to look at the video as it was being played. These are not easy to detect. And there's no natural light. These are the lights that the UAV has. And as it moves, sometimes these are actually one of the better quality videos. Most of the times as the UAV is moving around, it would kick up dust and so on. So that makes it even more difficult. And then it's about close to 200 meters from the surface of the ocean, right? So it's pretty deep. And they also have GPS and other, not GPS, but you have location information, but for various reasons, we are not making that publicly available. Okay. The next one, so switching the context, so underwater to above ground, right? This was a CVPR paper last year that we presented on methane detection from hyperspectral images. And as we speak, Satish, who will be graduating next in a month or two, he is trying to commercialize this with some help from NSF. So he has actually gotten some initial funding and has some investors. And the problem here that we are talking about is that detecting the methane gas from satellite and hyperspectral. And many of these things we didn't know when we started working on this project, but it's about 100 times more potent than carbon dioxide. And a third of it comes from the farms, a third of it comes from the oil and gas industry, and a significant amount of it from the landfills. So from the aerial observations, can you actually detect and localize and quantify this is the problem. So there are lots of people who are interested in actually doing this because of all the federal, whatever guidelines and rules and regulations one has to follow. So there's quite a bit of an interest in this. And the other side problem that he is interested in that he will be exploring is in terms of for agriculture purposes. So apparently, if you can measure some of these gas concentrations close to the ground, that can help the farmers. So that's the side one. In fact, he just got selected by the what is it called, the Smith Science Fellows. I don't know whether you have heard of that. So he's one of the Smith Science Fellows that was selected for this 2024 cohort. So that's a nice thing. It gives you, for those of you, are there any PhD students here?
Schmidt, right?
Yeah, Smith, yeah.
Schmidt,
Yeah. That gives you close to $250,000 for 2 years, and you can take that money and go anywhere in the world to do your postdoc. So it's like a good thing to try. So this is the problem, and each of these. Most of these things you need to be opportunistic when you look for problems. This one was not something that I wrote a proposal and wanted to do something. There was a local aerospace corporation that wanted to give us some money and said, do whatever you want, right? And then, but they didn't have the data for us to work on. So they said, why don't you look at JPL and see if there are some interesting problems? And we ended up, the end of the story here is that we ended up looking at the hyperspectral imagery, very poorly annotated. It's hard because the human experts is still looking at those. So what you're seeing on the left-hand side is a data cube that has about close to 450 channels, 5 nanometer separation in terms of the spectral thing, and could be easily anywhere between 30 to 50 gigabytes of data for one image. That's the data. So all of this is actually now available in Bisque with the annotations that we have. Probably this is the largest methane annotated data that's curated. So it's available through Bisque today. Okay. And that's how the images look like. So you have, once you stitch them together spatially, they could be like 20, 30, 100,000 pixels in one direction and maybe like 512 to 1,000 pixels in the other dimension. And that's what you need to manage. So the paper, CVPR paper that we presented last year, and then the primary contribution or innovation here is that deep learning cannot still benefit from signal processing. So if you do a good initial job in terms of extracting the right kind of features and then developing the machine learning models on top of that, you are likely to do better than not doing that. The primary reason here is that we have some prior information, prior knowledge. The learning methods, if you want them, those things to be discovered from the data alone, it requires lots of samples. So you can cut down on that space of how many trains and annotated label points that you have by bringing in whatever prior knowledge, physics-based or otherwise, into the. So in this case, traditional 1980s matched filters kind of stuff that kind of separates out the signal from noise and then building on top of that is what you have. Okay. And here are some of the examples. So these are actually pretty good in terms of. Left-hand side is the ground truth that's curated, and what you see here is so these are the methane plumes that you want to localize and identify, and it's doing a pretty good job here.
Can I ask a quick question with
Sure,
that?
sure.
So the data cube is itself 2D and then frequency in the third dimension, or what?
Correct.
Okay,
Exactly. So each one of the slices is think of this as a 512 by 512 image at whatever the, let's say, 400 nanometer resolution.
I see.
Right, and then so increment that on both directions. I think it starts from around 400 nanometers all the way to 2500 nanometers.
Okay.
And I believe the methane part is somewhere close to 2000. But you cannot, okay, so I mentioned about one of the innovations, the matched filter part. The second part here is that these are very noisy signals. I mean, the signal to noise ratio drops drastically. So the same methane plume, if you image it, let's say over an urban area, if the same thing happens in a rural area, the signatures look very different because of the contextual information impacting that. So one of the things, again, I didn't get into the details of the technical details that are in the CVPR paper. We are actually semantically labeling the surrounding regions using the contextual information to enhance this. So that really is.
Is this going back to the imaging, right? So essentially, is this some kind of reflectance-based imaging?
Yeah, basically, yeah, there's no active sensing here. Meaning there's no radiation that you are kind of, illumination that you are, it's basically with the observations that are reflected.
This
And-
here.
Yeah, and then so the sunlight also impacts that because when something gets heated, right, the surface gets heated, it has a different nature for the same ground level observations, right? And getting the ground truth is almost right? But one way to detect, for example, and then also hyperspectral imagery is very expensive because you essentially are flying an aircraft with the instrument and then you need to scan the surface, right? So it's also not easy to collect the data. In fact, I have a slide. I'll come back to this issue. But hyperspectral imagery is expensive, noisy, but it has the right signature, and you can use this to detect where the oil refineries or wells are there because most of them are associated with with the emission of methane gas. So some examples, I'm just going to skip through this and again stop me. So in general, what you see here is that we are doing a fairly good job, and that's what excited him. But what he wants, so scalability is an issue, scalability in terms of the imaging process. So what Satish and others are looking at right now is, can we extend this to traditionally available satellite imagery? We don't have any specific results at this time, but the initial results are quite promising. So hopefully we'll have something on this. So compared to the hyperspectral imagery, which are not satellite based, so you have to actually do the runs to collect the data. You do have this Sentinel and running data more frequent intervals. So actually you can do a much better job, at least in terms of the data collection, whether you can do a robust job to do the methane. That's the direction that we are having to take. And okay, so this is probably a time to play the Bisque module. So I don't have audio for this one, so I have to look at what's happening on the screen and try to explain. So what you're looking at here is that one of the pieces of data that's loaded, only one band is kind of, the RGB version is kind of shown here. And you can do some simple things to highlight the segmented regions. So that's the methane detector version. By the way, this image itself, probably you can't see the details here. It's this tens of thousands of pixels in one of this Y dimensions. X is fixed because that depends on the number of bands that you are looking at, or the sensors that you have on the spectrum work. Okay. Now, jumping to another topic now. So from methane to more on the brain imaging side. So this has been another one of those unfunded collaborations that has been extremely productive. So this is the joint work with Professor Jefferson, who is a neurosurgeon at UC School of Medicine. And what he's specifically, when he came to us, I don't really remember how that initial connection happened, but he was interested. He's been working on this particular problem for a long time in clinical practice. Identifying or detecting the condition known as normal pressure hydrocephalus. What it basically means here is that, in my non-medical terms, right, the ventricular region gets expanded, it blows up, and that creates symptoms which are very similar to Alzheimer's and dementia and a host of other kind of conditions, and that makes it difficult for them to detect this. Now, why do you care about detecting NPH, right? Apparently, this is something that's completely reversible if it is detected earlier. But it's often kind of, because the symptoms are very similar to Alzheimer's and others, they don't identify that as NPH, and so proper corrective actions are probably not. So about close to 1 million people have NPH in the US, and he's a neurosurgeon. So the way, if you look at YouTube and look at what happens to what's the procedure that's done, basically they will insert something here and drain the fluid into inside of the body, and that reduces the cerebrospinal fluid within the ventricles, and that kind of helps relieve the problems that you're looking at. Now, what is done in this particular case, so Evans index is one of the things that could be used, but by the way, this is not clinically used. So clinically, you look at it, if the symptoms match and the intuition of the surgeon matches the condition, you go ahead with the procedure. I told him it's very scary thinking about it like that, but that's the state of the art, right? So and some of the more quantitatively attuned doctors, what they would do is they look at this the section of the brain where this is the maximum, the width of the brain is the maximum, and then look at what the distance between the horns of the ventricles. And that ratio, if it exceeds a certain number, and this is all like, again, based on ad hoc things, right, or heuristics, if you want to say that. If it is more than 30%, then you think that, okay, this is potential candidate for it. That's what is being done. So we have the only ground truth that you can have is after the surgery, if things improve, then there is a correlation. But other than that, I mean, again, medicine is more of an art, right, than science even today. So what we did, again, not getting into all of the details here, but we have a fairly good prediction on the NPH side, which seems to be as good as whatever the ground truth is in terms of the, so it's about 80 to 90% accuracy. So it's not 100%, but it's very close to being what the doctors can do. Okay. And then again, the details are in a paper that's referred to, but this is one of those things where you bring in segmentation followed by some kind of an inference method to make that part. But the more interesting thing that we were all excited about, by the way, this is also in Bisque. So if you have the right data set, upload it, a DICOM anonymized subject, that's how Dr. Chen is using that, you can get a prediction. So that's currently inside of the.
Can I ask you a question?
Sure, please.
So yes, you can do those measurements, but this is MR, right?
This is CT scan.
It's CT, you're not doing MR.
No.
Suppose you do MR, you can
Yeah.
find the proton density. Why not use the proton density to figure out, the presence of edema, which is probably causing.
This is not edema. No, this is nothing to do with edema.
But there's water retention, right?
No,
There's no water. Okay.
I'm not a doctor, right?
Yeah, but I'm just
So
thinking.
basically, the observation here is the enlargement of the ventricles.
Only the ventricle.
That's it. There's nothing else. There's no, by the way, NPH could be caused by some kind of a, trauma.
Oh,
Right,
I
the
see.
head.
Okay.
But I forgot to mention, this is something that happens in a significant number of adults over 70 years. So this is age related, but on the younger side, of course, of the population, maybe because of the trauma,
Okay.
etc., that could happen. But there's no other.
But this must be contrast enhanced CT because CT itself won't do this. So it's probably.
This is. I don't think so.
Okay.
This is a standard CT scan.
Just standards.
Standard CT scan. And you can take a CT scan and drag and drop it and you can do it. By the way, these are all 14 bits, right? So 16-bit data, so you don't really see all of the details in a presentation slide. So, but the more interesting part here is that, and this is what we were kind of excited about, which is also available on the, this is the neurosurgery paper, is that you can do a pre and the post, upload it and actually get quantitative measurements and how much of the fluid was drained. Now, it looks trivial if you're thinking about this as a pure segmentation problem and then computing, but the problem here is that these brains, the images are taken at different points in time. Potentially different scanners, etc. So the normalization across, even though it's for the same subject, across imaging conditions is, and we have more detailed data available in the paper. So he's quite excited about this point. It's the first time any such quantification has been done, and so we are looking into what the next steps are in doing this. The next topic is switching gears here on image forensics. We all now know deep fakes is everywhere, right? But some of this work started much before the deep fakes became really popular, back in the 2010 or 2011 when we started looking into this, essentially trying to see how we can distinguish between what is real and what is not real, right? And that's a very hard problem. And there was a DARPA program that we participated in called Media Forensics or Medifor, and some of the methods that were developed here were part of that. The bottom line here is that we have now a forensics tool in Bisque. Now, how well it works depends on the context in which you are using it. And we wanted to we wrote a proposal to NIH, saying that, and this was 2014, that it might be useful to have a database where you can routinely screen these things, etc., right? And the reviewers, and this is a literal comment from one of the reviewers, but why do we need this for biomedical imaging? We don't have this kind of an issue to detect manipulated images. This was a literal comment from a reviewer. And this was 2014. And since then, at least 3, 4, I don't know, there was Stanford president, and there were a few others who were impacted. There was a research issue and all. So it's not as negligible as the reviewer's comments indicated, but it's still it's still a problem, and it's become worse now. In the 2010s to 2014 timeframe, there were no AI tools that are available. Today, A high school kid can take one of those apps and manipulate these things and you won't tell the difference. It's pretty sophisticated. In fact, this was something that we did long time back, but I just wanted to, for those of you who are not familiar with this forensics part, it's very easy to change or manipulate your influences. So here is one example, but you probably have seen the Tesla thing, right? Most of you are familiar with that. So it confuses the Burger King ad to be an exit sign up because of the trend that might have happened in terms of people taking the exit when they see a Burger King advertisement. But on a more serious note, right? The bottom line, again, you might have seen examples like this before. So that one, pick any of your classifiers, it gives you 100% or close to 100% saying that that's a ship. And same with the dog. But if you give me, if I have enough information, I can actually take that image, manipulate it the minimalistic part in the smallest possible way, and make this this to be inferred as a dog. And you can do that without significant effort, right? So that's something that anybody can do today. So it's not rocket science, right? And in terms of manipulations, you can also do, there's lots of sophisticated methods that are out there. So for example, here on the left-hand side is the original image. Again, these are all taken from the publicly available stuff. It's nothing to do with our group. But the part that we did was, in fact, the manipulations. You can say that you can actually identify regions you want to delete, regions you want to not touch, etc., and create something that looks pretty realistic with the person wants from the image, right? So these are things that you can do in a few seconds' time. The methods that we have that were kind of, I think still is state of the art is, I can actually identify where the manipulations are. So that's on the detection side of things. There are two satellite images here, or aerial images. I'm not sure if satellite or aerial, regular pictures. Do you know which one is real here? Left or right from your direction? Any guesses? Which one is real? How many go for left? That's the problem, right? So there are actually a couple of things that sorry, go ahead.
No, the bottom left corner.
This part here?
That part, yeah.
This one. So the right one is the real image.
Yeah, it looks like.
And the left one, you see that the lake is replaced by some landscape, right? So by the way, this editing is done actually not in the image space. So I took a map and edited the map. So there was a lake here, and I removed the lake body. I wanted it to be replaced by whatever that looks similar to the ground. And so you can think of different ways of editing those images too. And one could detect it if you have the right tools. So we have a couple of state-of-the-art tools inside of Bisque. One coming from USC group. That's the Mantra Net. That's I think open source public one. And something that we had developed on the SIM carving detection. So in case you're interested in playing with that. And I think I'm running out of time here. How much?
Yeah, I have 5 minutes or something. I mean, we have a lot of questions, so.
Yeah. So I'll skip the microstructure part because this is again another interesting materials related application where we can actually synthesize the images. So all of these, what you see in the next few slides are currently inside of this. I'm going to skip it because I would like to get to the most recent project that's Smithsonian related. One quick mentioning here is that just like medical imaging, hyperspectral images, getting these material samples is very, very expensive. A single sample of your 3D volume can cost tens of thousands of dollars. What you see is an instrument that was developed at UCSB by Professor Trisha, and it has 3 different sensors. Essentially, you bombard the material with electron beams and look at the scattered information, and from that you can try to compute the underlying crystallography information. So that's the material structure part, and there are lots of interesting applications of that. If you can actually go from, so the thing is that you cook the data, right? Cook the data materials. That results in some structure which has certain properties. But Ideally, you'd like to go the other way, right? You want to define, I want this material with these kinds of properties. What's the best ingredients that I can cook and how I can cook that so that I can get to that. So the intermediate step is, I think there's a good understanding that structure relates to properties, but how can I get the structure available from the material? That's the holy grail of many of the researchers for the researchers in this space. So let me skip this. And this is something that Tanya would be interested in. So this is a paper that will be presented at CVPR next month by, again, Satish is involved in this. And this has been a great collaboration with the Smithsonian Institution. So they came to us interested in, so what you're looking at here is basically an aerial image, a drone or whatever. I don't know how this one in particular was taken. And you want to detect and count animals of interest. So at a very high level, that's the problem. That's of interest. And so this also has the connection to the researchers in Kenya, because they are actually, we went there and gave a couple of days of presentation on Bisque, how to use Bisque so that you can upload the images and participate in the annotation process and do the detection and the census part. But we almost didn't have the paper submitted to CVPR, meaning after it was accepted, because we said we will make the data available, but we still haven't gotten the intergovernment agencies to sign off on it. They have permission to still go ahead and have the paper submitted, but we're still working on making this data available. We have the data, but it's not coming to access.
Are Kenyan researchers part of the co-author list?
Yes, they're almost all of them, including the pilot who flew the thing. They're all part of this, but still a challenge. Because I think for them, the data is good. And I think there were some previous instances where probably things were not done the right way that might have impacted them. So I think they've been extra careful. And again, when you put two government agencies to work together, usually anything happens, right? So those are the challenges again, right? They have large amounts of data. All of the things that I mentioned at the very beginning of the presentation are still valid. And again, I'm going to skip this part. The main contribution here from the computer vision point of view is that it's like a needle in a haystack kind of a problem. Your footprint of these animals are going to be really, really, really small. So the signal to noise ratio when you're analyzing the large image is going to be pretty low, right? And right now the state of the art is that, this is literally what I was told. So the pilot is flying the plane, and then the cameras are looking in maybe just looking downwards, right? And then somebody looking outside and counting. I mean, that's the kind of, and may have some additional tools when they go back and do something, but that's the ground truth, because they're not making up the images. But these were the older days, maybe five years back. So I don't know what today is available. Maybe Tanya has more information than I do on this one, but this is an interesting problem from a computer vision side of the things, right? And so what we try to do is to actually enhance the signal. And in this case, so just like if you remember the methane mapper problem, where we had the match filters here, you want to separate out something that is common versus something that's not so common. So some kind of a high pass emphasis of your signal would lead you to better classification and detection. So that's kind of is being shown here. So you're focusing your attention on areas of interest from these really large images so that you can do a better job on those focus regions. Okay. And I think there are some examples of the detections and so on. And given the time constraints right now, I'm going to skip through. So the method that we have is really good at this point, at least as of now, right? If you look at all of the other publicly available methods that you can use to train versus what we have, we are getting at least a performance improvement of a factor of 2, which for those of you who are not in computer vision, even a 1% improvement is great. So a factor of 2 is not bad in that sense. And both the detection and classification things, we are the state of the art. Okay. I think it's too late to ask this question, what is this, what is this and that, right? But I wanted to kind of close this with summarizing what you have seen so far. And you can store, you can visualize, you can economize, but I think something that's missing, and I asked my students to put this slide in a better way, but they didn't have the time. The collaboration is the most important part. Collaboration, sharing and collaboration. So all of these things that you can do out of the box today, okay? And I have a number of videos which will not have time to play in terms of how one can do the annotations, correct the annotations, etc. So I'm going to just describe that after this slide. So this is the way on the back end, the hardware side of things, right? You have the front side is the web browser. Back end are your whatever compute clusters. We don't have significant resources at UCSB. Cybers had a much larger capacity. I think Nirav told me once that they may manage about close to a few petabytes of imaging data. And then the image part of that was this supporting that. So you have a storage service that manages all of the storage part of it, and then a Kubernetes cluster that kind of schedules the jobs together with the Argo workflow. So all of these things are currently nicely integrated to make this happen. And you can take this and run it on, for example, Amazon or a Microsoft Cloud service today. Okay. And I think I talked about pretty much all of the features here. I did mention 3D data, so let me see if I have any examples of that. Okay. So you can do all of these things from within the browser. And what we don't have, it's hard to annotate the data in 3D, so you still have to depend on a slice by slice annotations, but anything in 2D, you can annotate quite easily. Okay. And then let me go back to the. Oh, yeah. So while on this one, so the images are in any dimension, there's no limitation. You can have a 1,000,000 pixel by 1,000,000 pixel image. It depends on the computing thing and then how fast you can push that to the client side, but it's more of a scalable thing. So think of Google Maps, right? When you zoom in, it gets better and better. So something similar to that is available here. I think I have touched upon most of the things that I wanted to. The last part of it here is, yeah, so these are the videos that I talked about. I'm going to skip through all of this. You can do so one thing that we recently did is so this like in its current version, it actually gives you some very simple Photoshop kind of abilities, enhance, do things, draw bounding boxes, annotate. So all these things are considered as graphical annotations, the objects that you can actually push that to, let's say if you have a machine learning training. But what we did recently is that if you have a method, so let's say that you have the best possible sheep detector, you can bring that in, put that into Bisque, and then depending on how that's engineered, might take anywhere between a few hours to a couple of days. But once you have done that, you can, once you have the detector in place, all of the detections will become automatically graphical objects that you can actually then go and correct, update, if the things are not good, delete, etc. So all those interactive things you can do today. Feel free to ask me questions. I think I'll probably at this point just leave on this slide here. The direction that we are pushing is that can I go from the images that are uploaded to whatever modules, but complete the learning cycle? Meaning something like an active learning for those of you on the machine learning side. So that's something that we are working on without leaving this. Can I train the models in a way that we don't want to put everything inside of Bisque, because that's also then becomes a monster that's not manageable. But how can you connect that to training methods that are close by so that you can have a good two-way communication between once I have updated, push that to my training service, maybe another platform for the TensorFlow, update and put the modules back. And that I did not do any computational module one has, you can put that as a module, as long as that requires, the imaging data to be analyzed. So we can, and I think we should thank the localization process for that, because that has made our life extremely simple. So these are some of the things that we are working on today, including the 3D annotations, what's to do, how best to engineer that. And then, last comment, anything that you do in Bisque is the provenance of your computations are maintained. So you know exactly who has done what. So if anything that was done 10 years back, you should be able to reproduce those computations today with exactly the same results. So not only the data is associated with the runs, and I didn't get a chance to show you all. So anytime you do let's say process an image, the output of those computations, the method that's used to do those computations and the data, everything is maintained the way you did that experiment. So you can go back and redo those exact same computations. So I should have lots of others, but thank you again, and happy to answer any questions.
Yeah, so there is one question, Elmar, on the Zoom, you can unmute yourself, ask the question directly.
Hi, I wanted to ask, given the capabilities that you've enumerated, and I guess what I'm particularly having in mind here is not so much the image processing pipeline, but the annotation, segmentation, and object identification capabilities for curating annotated data sets. Is how would Bisque compare, or alternatively, if it doesn't really compare, to CVAT, which I understand is a tool that's commonly used for that in machine learning.
Right. In fact, we have a CVAT integration. CVAT is more of an annotation tool, which has now evolved into bringing in maybe many of the machine learning methods as part of the annotation. But we do have CVAT integrated. We have video where we found that's particularly useful. Is CVAT does some of the tracking and things like that that we could kind of leverage on that. So we are thinking of CVAT as another module within Bisque. We are not directly kind of we're not in the same space, right? CVAT doesn't do all these things that I mentioned, like provenance sharing, all of that stuff. Maybe one can engineer it to do that, but out of the box, it doesn't do that.
Okay, are there any other questions?
So expanding sort of on Hilmar's question and more broadly, how does this maintain the connection to metadata, and particularly the metadata that comes from not directly, but as part of the process of interacting with, if you use, for example, if you use CVAT, you're producing annotations, which are.
You can push that back into Bisque with the annotations.
So the boxes and all that. So,
Again, that might be still in experimental stages, but you can see actually when you go to the Bisque page, I don't know whether you can see that without logging in, there is a CVAT box. You can then, when you click on that, the data gets downloaded onto your native, your local client-side computer, and you can do your annotations, and then you can push it back. So the annotations and the data, with including the graphical annotations, will get pushed back into this.
And so see that as an example, but there are plenty of other, and where you can push metadata back. And so then the expanding that even further to multimodal data. So let's say that I'm working both with a habitat data from remote sensing as well as, animal detection. Then I have other animal data, image data sources that are coming in, sort of all connected. And I want to tell it how it's connected.
Right.
Yes.
Great question. So a couple of things that are kind of embedded in that question. Anything that's very specific to images, right? Like CVAT is a good example, right? Where you are localizing something and saying this is XYZ. You want that to be associated with the image and the region locally, right? So those, we do that, right? Now, if I take an image and then I say that there's a lot of descriptive or other metadata about that image as a whole, okay? There are two possibilities at that point. You can push that as a metadata, right? But maybe this other thing that you're talking about is not just metadata, but a completely different piece of data related to this original image. And I'm just thinking here, maybe you can say that this is the same subject, right? I have the MRI, fMRI, maybe different kinds of things, but I want these things to be linked together. Maybe that was part of your question.
Mhm.
If that's the case, so this provides a data type called data set.
Mhm.
So you can take any arbitrary combinations of the data that you have uploaded, either it's based upon, let's say, PIs who have done the experiment or something else, maybe geographic location, say, these things should be together as my Manjunath data set one.
Mhm.
So that's another, but they're not directly connected, but they're part of the data set, and then you can maybe whatever methods that could potentially use this data set can push.
Or the other way, yeah.
Exactly. You can.
Okay.
Other online, there looks like there's a lot of comments
No, no,
online.
I am reading that. I think those
Oh,
are all discussions.
Okay.
You are seeing better. I am monitoring. Okay,
During this part.
There are no more questions. Okay, so let's give a big thanks to. We'll conclude here. Thanks a lot again for all in person as well as.